So, I just went through my whole spiel and I wasn't recording, so hopefully it'll be better. So I was looking at your email here. I wanted to talk through each of these little points briefly, just so you know my thoughts on them. So this moving away from my poster, one thing that Zach had mentioned, Zach Simmons, when we were doing thesis presentations at the end of last semester, is he said that he wanted to make sure that people who didn't know anything about neuroscience could still follow along. Additionally, with peer reviews that we did in class, a lot of people said, as soon as you said this word, like nucleus accumbens, I was lost. So I'm trying to make it as simple as I can, especially for OAS and for Founders Day, because you say the word neuroanatomy and people tune out because it's something that they're unfamiliar with. So that's my purpose of, I guess, moving away from the poster. I'll try to get more background this run through. I did look through this to get some ideas of what you were talking about. I'll add that in. I added in the timeline. I'm not exactly sure what a glass brain is. I assume it's those photos of transparent brains with the region inside of the brain that's like a brighter color, or it's a darker color than the cortex of the brain. I couldn't find anything that I really liked, especially a mouse brain, especially the nucleus accumbens and the anterior cingulate cortex. So I'll continue to look. I only looked briefly, but yeah, I couldn't find anything that I thought was good. So with the graphs being accompanied with photos, one thing that was said to me when we were doing these presentations in class, in thesis class, was these photos need to be clear right before I talk about the graphs. If you think that I should have another photo of the brain up here, or a different view of it, then I can add that in. But this was my whole purpose. This is the purpose for this graph. Same with this one and this one. So they go together in that way. But I can always add more. And then, yeah, I'll try to focus again on that. Maybe even saying this at the beginning, this is why we're looking at the accumbens core and shell. But yes, I'll try my best to see. Hopefully this works. Hopefully I am recording. I think I am. But let's go. Round 2 again. Okay, slideshow from start. So, and I haven't practiced since I looked at since I did my other presentations, so this might be a little rocky, and I might say some things that I will not say when I'm presenting, because I will be practicing more and more. But anyway, here we go. Thank you all for joining me today to talk about Huntington's disease and ask the question if dopamine signaling changes in a brain with Huntington's disease. My name is Ruby Perez, and I will be representing the Pitzer Lab from the University of Portland, where Zachary Daniels and Dr. Mark Pitzer and myself spent many months working to answer this question. Before I started this research, I had no idea what Huntington's disease was. So I think it's very important that we all start on a simple background of what Huntington's disease is, just so we all have a basic knowledge that helps us understand why we did what we did in this study. So Huntington's disease is a neurodegenerative disease. It is fatal. Huntington's disease, it follows an autosomal dominant inheritance pattern, meaning that if two parents have children and one of the parents has the condition, in this case the father has the condition, every time this couple has a child, the child has a 50% chance of inheriting the condition. If the child has a mutated Huntington gene, then they will show symptoms. This moves into my second point here. The Huntington gene is the gene that is problematic in Huntington's disease. And what happens is the Huntington gene has the 3 DNA letters, CAG, repeating over and over again. But when it becomes problematic is when these repeats, the nucleotide repeats, exceed 27 CAG repeats. When it does exceed about 27, and again, there are, there are massive human variability with this, so you might have 28 and you might be fine. It depends. And, just like anything in science, it depends. But when you have an abnormally long Huntington gene, you get what are called inclusions. And inclusions, maybe I should have a photo of inclusions here. Inclusions happen when the gene is so long that it folds itself over. And it's easy to think about this like a string with beads on it. And you can easily put, let's say, 27 beads on this string, but as soon as you start to cram on more than 27, it gets a little tight, and there's nowhere for the beads to go, so they fold in on themselves. So this protein then becomes misfolded. The gene is misfolded. This causes the gene to be sticky, and when this happens, it becomes toxic to the cell. And so in the brain, you've got these clumps of this Huntington gene that are toxic to the cell, which ultimately causes neuronal cell death. Neurons in the brain are dying, and because of this, we have a massive atrophy and shrinkage of the brain, causing symptoms. The symptoms are classified into 3 categories. We've got cognitive symptoms here on the bottom, and these include things like dementia and problems with working memory or short-term memory. The motor symptoms are the most common, most studied, most understood symptoms. Maybe some of you who are familiar with Huntington's disease have heard of Huntington's chorea or just simply chorea. This is classified as involuntary motor movement of the limbs. There are things that happen as well, like facial grimacing, problems swallowing, problems breathing, things along those sorts. What is lesser known is that Huntington's patients have psychiatric symptoms that happen before motor symptoms. And the motor symptoms typically are the marker of when people go into the clinic to be formally diagnosed with Huntington's disease. So if we can find out what's going on in the brain that are causing these psychiatric symptoms about 10 years before motor symptoms show, there might be something that we can do to help these people sooner. There are a wide range of personality deficits that happen and psychiatric symptoms that occur, but the ones that the lab was interested in were impulsivity, risky decision making, and the perseveration or the continuation of these maladaptive behaviors, specifically impulsivity and risky decision making. So we're looking at the mesolimbic cortical pathway, and we're looking at this pathway because of 2 nuclei in particular. We have the nucleus accumbens, and here is circled the prefrontal cortex, but we will be looking at the anterior cingulate cortex, which is just a little closer to this structure here, which I believe is the corpus callosum. Yeah. Both of these regions of the brain receive dopamine from the brainstem here, from the ventral tegmental area. And the nucleus accumbens and the anterior cingulate cortex mediate impulsivity, risky decision making, and perseveration, or perseverative behaviors. So we weren't looking at human brains, we were looking at mouse brains. And Huntington's disease is a purely human disease. There is no mouse Huntington's disease. So why can we use this mouse model? Well, mice are genetically similar to humans, making inferences possible. And this mouse model here is interesting to us because it has three copies of the Huntington gene, two of which are its own mouse Huntington genes, one of which is a human Huntington gene that has been expanded to 82 repeats. This mouse model, the N17182Q, shows similar symptoms to adult onset Huntington's disease. So, from all of this, my hypothesis became that those with Huntington's disease, the mice with Huntington's disease, will have less tyrosine hydroxylase, or dopamine, in the mesolimbic cortical circuit as the disease progresses when compared to their healthy controls, or wild type. So I had just said tyrosine hydroxylase in that previous slide, and you might be like, oh, what did I miss? You didn't miss anything. I'm stepping ahead here. We actually looked at tyrosine hydroxylase, and tyrosine hydroxylase is here in the green circle, and it is the rate-limiting enzyme in dopamine production. So it is an indirect way of measuring dopamine. But the reason why we wanted to measure tyrosine hydroxylase is because it is more stable over a longer period of time than dopamine is. Dopamine degrades pretty rapidly, and tyrosine hydroxylase does not degrade nearly as quickly as dopamine. So we turn to tyrosine hydroxylase, but I will be saying dopamine just for the ease of syllables through my presentation. So the methods here can be seen pretty clearly on a timeline. So down below is a table of the groups that we had, and they will become more clear, hopefully, as I go through this timeline. So we start with birth. All of these mice, all of the mice were born at the same time, and there were a total of 31 of them that we used in the study. We then sacrificed half at 5 weeks, and we sacrificed the other half at 15 weeks. And half of those two time period groups were split into either the wild type control condition or into the mutant condition that have that extra mutated Huntington gene from humans. Then we treat the tissue, which takes about a few days to a week, using standard immunofluorescent or immunohistochemical techniques. And then we took some photos under the microscope. The camera was positioned above that allowed us to look at these photomicrographs. And this is what we saw. We then transferred these photos to a computer that allowed us to measure dopamine intensity. So we're looking at the brightness here, and the brighter something is, the more dopamine is there. Except for, you might be like, oh, well, this region right here is really bright. Another thing that is really illuminated in this process is myelin fatty tissue. So right here is a commissure that connects the front of the brain to the back of the brain. So this is not dopamine, this is a connective tissue. That might be wrong. Please tell me what I should say there. So moving on to the brain regions that we looked at. So I had mentioned the nucleus accumbens. Well, the nucleus accumbens can be broken down into two subregions, the core and the shell. So here we're looking at the core of the nucleus accumbens. And the core is responsible for recruitment of motor regions to physically go toward a stimulus. And it has been labeled as the go-to nucleus of the nucleus accumbens. Additionally, I will mention that the striatum was also looked at, and the striatum is part of a different pathway, the basal ganglia, and the striatum is also a motor region that recruits other motor regions to physically move. So here we're looking at motor movement, kind of these that second category that I had mentioned, the motor symptom category. So again, we're asking the question if dopamine signaling decreased. No, we didn't see anything really interesting happen here. This red line represents the mutant condition, and at 5 weeks, the mutants aren't any different than mutants at 15 weeks. Additionally, the controls, or the wild types at 5 weeks, are not different than the controls at 15 weeks. And further, the mutants and the controls are not different than each other. The same can be said in the striatum here. The wild types up top here in blue at 5 weeks are not different than wild types at 15 weeks, and the mutants at 5 weeks are not different than the mutants at 15 weeks. And again, further, these two are not different from each other. So nothing interesting happened. Moving on to the region that we had said was part of this prefrontal cortex, the anterior cingulate cortex. So this is involved in these higher cognitive functions. We've got decision making, emotional regulation, and error detection. This is what the anterior cingulate does. And you can see here in yellow, this is where the anterior cingulate cortex is located, right above the corpus callosum. And yeah. So again, asking the question if dopamine signaling decreased. Yes, it did here. So we saw that while there was no difference between mutants at 5 weeks and wild types at 5 weeks and wild types at 15 weeks and mutants at 15 weeks, there was a difference between those at 5 weeks and those at 15 weeks. You might notice that it looks like the mutants are on a more rapid decline than the wild types. I will mention this and what I think is going on later. But we saw a main effect for time. So there's something about 5 weeks that's different than 15 weeks. Something is happening in the brain. Moving back to the nucleus accumbens, we're now going to look at the shell. We talked about the core, now we're talking about the shell. And the shell, again, to remind you that this central outline here around this anterior commissure is the core, the go-to motor nucleus of the nucleus accumbens. Out on the periphery is the shell, and the shell kind of acts as this brake system, suppressor of behaviors that are being sent from the front of the brain, our higher cognitive functions that's saying, hey, you shouldn't be doing this. The shell is like, yeah, you're right, I'm going to stop. So what did we see here? This might relate to something like impulsivity or taking risks. And we saw dopamine signaling decrease. And we saw it, again, in an interesting way. So we saw a main effect for genotype. And what this means is there's something about being a wild-type mouse that makes you different than being a mutant mouse. So there was no difference between 5 and 15 weeks, but there was a difference between being normal and being a mutant. So it appears there's less dopamine signaling in a mutant. They start out with less and they end with less in this time frame that we looked at. But again, time will be discussed further in the discussion. So to wrap up again, there was nothing really interesting in the two motor regions that we looked at, the nucleus accumbens core and the striatum. Nothing happening there. We might not be looking at the right thing, though. In the anterior cingulate cortex, I had mentioned that it might seem that the mutants are declining at a faster rate than the wild types. I will further talk about this in the limitations of this study, but there's nothing that's saying that this degradation is due to normal aging. However, in the shell, this is where we found our interesting significance here. So to further say, the mutant mice had less dopamine than the wild types in the shell. And the shell is kind of responsible for impulsivity, risky decision making, and perseveration together. It has these three components along with the other regions that we looked at. But there's some possible explanations as to why we saw a decrease of dopamine in the shell that was significant. So So with impulsivity, it has been shown that a decrease in dopamine within the shell has been correlated with an increase of inappropriate responding. People become impulsive to act, or they're quick to act. So we're all here giving our presentations. An example of this might be, if you had less dopamine in your shell, dopamine signaling in your shell, you might exhibit behaviors where someone calls your name to give your presentation. You walk up, but you walk up too soon, or you take the microphone from the presenter too soon. You're being quick to act. Additionally, I'm not sure how many of you are familiar with Woody Guthrie. I wasn't before I started this research, so I don't expect you to. However, he was a famous country or classic rock singer. I don't know what he is. But there have been, and he had Huntington's disease himself. There have been numerous encounters of him being impulsive, and one in particular that I'm thinking of is he was at church one day, and they were passing the collection basket where people put donations into the basket, and the basket passed by him, and he put the money in his shirt. And we all want money. We could all use more money, but he did this impulsive action, and it might be because he had less dopamine in his shell in the nucleus accumbens. With risky decision making, risky behaviors, the shell plays a role in learning to inhibit behaviors associated with punishment. And an example that I think of when I think of this is, let's say a Huntington's patient goes to Vegas and they sit themselves down in front of a slot machine and they start putting their money in. If the shell is inhibited, if there's less signaling, less dopamine in the shell, these people with Huntington's disease are not going to know, they're not going to learn to stop. They're being punished by losing money, but they're not going to learn that this is bad for them. They understand, they don't learn. And then finally, with perseveration, a decrease of dopamine has been correlated with behaviors continuing after reinforcers and punishments have been removed. So again, these people with Huntington's disease just continue to make these behaviors. And that is correlated with a decrease of dopamine within the shell of the nucleus accumbens. So here we are with the limitations and the future directions. I had mentioned time. First of all, time in that the time that it took to run this study, the time that was devoted to this study, could have been much longer. This could have easily been a 5-year study, maybe, but it takes many months to treat the tissue and to look at the tissue and analyze the tissue. But what would have been supplemental and made our results more robust is if we were able to run behavioral tasks on these animals, which we did not have time to do. This would have taken a long time. We would have had to rear and raise the mice themselves, teach them behavioral tasks in the short span of time that we have, 5 weeks, and make some inferences about their behaviors and their levels of dopamine. But it was not feasible within this study. Additionally, I had mentioned rates declining faster in mutant mice. This might be because we didn't have the ability to look at different time periods within these mice's lives. So within the anterior cingulate cortex, we're noticing this pattern of mutants' dopamine levels decreasing at a much rapid rate than those who did not have Huntington's disease, and it would have been interesting to extend that time period beyond 15 weeks to see where these two officially converge from each other. The problem with this is that these mice don't live much longer than 15 weeks. Sometimes they live to 20, sometimes, about 28 is when they all start to just drop off. It's very unfortunate, but that is a benefit to using this mouse model, is that their time is quick, not quick enough, not slow, not too slow, but it's kind of like the middle porridge that we chose. Additionally, with the shell, we didn't see where it made a difference in being a mutant that caused that was related to having lower levels of dopamine in the shell. So it would also be interesting to look at where exactly. Is it at birth, before birth, when you start to have less dopamine in the shell? With future directions, I think the most logical next step would be to look at receptors. So again, I have this photo here of a presynaptic neuron synapsing with a postsynaptic neuron. You can ignore the liver right now. But what we looked at was this neuron here, let's say neuron 1, releasing dopamine to neuron 2. What we didn't look at is neuron 2. We didn't look at how much receptors neuron 2 had. And it might be that the receptors are degrading, or it might be that there's a compensatory effect that's happening that's causing a significant change that we're not seeing because we're not looking at it. So with that, I think the one thing that I hope you take away from this presentation is that anytime you think about neuroscience or neuroscience research, Huntington's research, any research dealing with the brain, is to think of the brain working in a circuit, and this circuit has to be thought of as a whole. And so to better conceptualize this in simple terms is I have a simple electrical circuit here with a light bulb, a battery, and a switch. And ultimately the goal of this circuit is to turn the light bulb on. If the light bulb's not turning on, there are a few places that you're going to look. You're going to look at the light bulb itself. Maybe the light bulb is broken. You're going to look at the battery. Maybe the battery doesn't have enough energy to power the circuit. And you're going to look at the switch. Maybe you have a faulty switch. If all three of these regions don't show anything, then maybe you'll look at the wires. Maybe the wires, there's a break in one of these wires. Let's say this wire here is broken and that connection isn't being made. But with research in neuroscience, it's not always that clear. It could be that this red wire here is degrading just a little bit from the battery to the switch. But it could also be that in addition to that wire degrading, the wire from the switch to the light bulb is also degrading. And then the wire from the light bulb to the battery is also degrading. Not enough for them to be dysfunctional on their own, but in summation, causing these problematic things that we're seeing. With that, I have many thanks to give to my lab partner, Zachary Daniels, and my lab advisor, Mark Pitzer. I couldn't have done it without you guys. All those many months that we spent in the basement. No lights looking at mice brains. People might have thought we were insane, and maybe we are. Additionally, the McBride Lab at the Oregon National Primate Research Center associated with OHSU for the donation of these tissue blocks from the mice themselves, because I love animals, and this is a very sad thing to happen, but they were very much needed. And additionally, and finally, Dr. Zach Simmons, who is the psychological advisor for the department in helping the students combine their research into papers and presentations for people to listen to. Yay. But where is he helping me now? Nowhere. Okay. So that's that. Hopefully it was not longer than 15 minutes. Yeah.